I substituted, today was going to be chapter 8, which I think was biotechnology or something. And I substituted this one because we are going to be doing phage display, or really bacteriophages, or working with them on Tuesday, a week from Tuesday when we come in. So I thought this would be a better lecture to have before that. So this is going to be about mainly viruses. There's viroids and prions a little bit towards the end, and that's the part I can kind of just let you review on your own if we get too late and this doesn't seem like it's, the attention span is there. So I'm going to really focus on viruses, and there's viruses, and I slip often. I call a bacteriophage a virus. But there are two kinds of viruses. There are viruses that infect humans and prokaryotes, and there's viruses that infect eukaryotes and humans, and then prokaryotes like bacteria. So when it's a bacteria that infects or a virus that infects the bacteria, it's called a bacteriophage. And that's what we're going to be dealing with in the lab, is a T4 phage, in fact. So you can see viruses have lots of different sizes. This is considered a eukaryotic cell, so this would be like our host cell. And this is what a bacteria looks like, even compared to our host cell. It's pretty small. And then you look at the different viruses. They're very, very small. And this is the virus that we're going to be talking about at the beginning of the lecture. And also it's the one that we're going to be using in lab. And again, it's a bacteriophage, and this is a bacteria, and it's going to be infecting it. Okay, so kind of expect, just because of this, maybe some conversions, some conversion factors, may be a problem with converting from one thing to another, just because we have a lot of nanometers up here, so it's something that you should know. And then we have millimeters when we are going to talk about the phage display. All right, so viruses, this is really pretty interesting. The most abundant biological microorganism, we can't even see a virus. You can't even see it, well, you have to use almost a scanning electron microscope to see it. We couldn't see it with our microscopes. And yet, as far as abundance, you can see this drawing, the abundance is the most abundant organism on the planet. And yet, you can see by biomass, of course, this microorganism is very, very small. So by biomass, it's not very large, but the actual number of them. There's about 400,000 different species, they think. But we have only really discovered 8,000 of them, which means there's only about 2% that have ever been discovered. Viruses, the coronavirus took us a little bit by surprise. That was one that obviously is on part of this 400,000 that are different, and not one of the 8,000 that we already knew. So that being said, you can imagine how many viruses are out there that are eventually going to be discovered, and discovered in what way we don't know. Either tremendous disease, it could be a plant virus, it could be some other kind of virus. But there's probably a lot of viruses that we're going to be finding in the future. Virosphere, this just means how long have viruses been around, and they've been around since life has been on the planet. Viruses infect everything. They infect plants, they infect bacteria, archaea. So anytime there was life, then there was a virus. And the reason they have to have a life form is because they don't have any mechanisms within their own realm in order to replicate or divide or have any energy source. They don't have any membranes, etc. So we'll talk a little bit more about what they don't have, and yet the reason that they actually need a host in order to survive. So there are major costs of mortality, whether it's plants or whether it's other animals or bacteria, etc. It's because the only way that they survive is to basically use all the mechanisms of their host, and that usually causes their host to deplete its energy, and so therefore it starts to die. Or when they leave their host, they bust out of the host, and that's usually in bacteria. And when they do that, obviously they have to kill the host to get out. So mortality is a pretty high feature of viruses. And another thing which is very surprising is it's a major driving force for evolution. When viruses, not all, but when many viruses get into a cell, they will integrate into the genetic material of the organism. And then when they leave that organism, they take some of that organism's genetic material with the viral genetic material. And then they integrate into the next host that they have, taking not only their own genetic material, but a little bit of the genetic material from another organism. So that then allows some organisms to kind of switch around some of the genetic material, and it has been very beneficial to some organisms and some organisms not. But when they sequence organisms, they find a lot of this extra extraneous DNA in the organism, and they can't figure out where it came from, but they usually find it right next to an embedded virus, and that means the virus probably brought it in with them. Okay, so viruses takes their genomes from one and brings it to another host. So here are some forms of what viruses look like, and I have another slide that we'll talk about a little bit more. But when we think about viruses, whenever were viruses discovered, we knew about bacteria long ago. We know Leeuwenhoek when his microscope looking at the little animalcules. So that was in the 1600s. They really didn't find bacteria or say bacteriophage because it was a virus that actually infected a plant. So this was in about the 1800s, and the guy got a Nobel Prize for it in about 1946. It's because they couldn't see it. So they knew what it was doing, and they knew that it was infecting a plant, but they couldn't understand how it did it, and they also could not ever discover what it looked like. So that's why it took so long to get a Nobel Prize. And when they first discovered this, it was Pasteur that got involved in the very beginning. And then from there, he couldn't figure out a lot of the plant viruses and things like that because he just didn't understand. He understood a little bit about bacteria, but not about viruses. So finally, this virus that infected the tobacco plants, they ground up the leaves of the tobacco plant. And you can see it could make some kind of a slush. And what they did is they filtered that, and they used a fine filter that had holes that bacteria could not go through. And what was happening then as fluid would just go through. So in the bottom of the flask, they would have what they thought was just fluid. And on the top, they had what they thought was the bacteria that was causing the plant to die, shrivel up and die. And they looked at the bacteria, they tried to infect plants with it, and nothing would happen. So they finally said, well, let's just take whatever's in the bottom and see if we can have that work. And that also did not work. And they couldn't figure it out for a long time until they realized that they had to have the virus and the bacteria together, because whatever this was, this living thing in the solution had to have another host in order to survive. So it took a long time to figure out exactly what was causing this curling up of these leaves and making the tobacco plant pretty much useless. So once they figured this out, they really didn't know how to combat it because they couldn't see it. They just knew that there were certain bacteria that would get infected with these mysterious things. They couldn't grow it on agar plates. They couldn't look at it. They just didn't know what it was. And they didn't have sequencing at that time, so they just figured it was something that needed a host to survive. It's some kind of a parasite. All right, so from there, I guess, 50 years or so later, they did figure it out. But at the very beginning, they had no idea. So viruses, they have a lot of different shapes. This is what the tobacco mosaic virus looked like. You can see it's kind of a helical rod shaped. Here's something that's a little bit fatter. So rabies, and then we have different viruses in different shapes. These viruses, when you see them up there, all you see is this is protein, and there's a protein coat around them. This is all protein around this. There's all protein around these. There's all protein around these. This one has an envelope, and it has also more protein. So within the interior of all of these, interior here, 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 and here, they have the genetic material. So you can see that this is just protein and genetic material. And that's what a virus is made up of. It doesn't have any kind of organelles in it. It doesn't have a ribosome. It doesn't have cell walls. It doesn't have a mitochondria. It doesn't have a cytoplasm. It has nothing except its genetic material and some protein that kind of makes up a coat. So if it doesn't have ribosomes, what does that mean? What can't it do?
Make protein.
Can't make proteins. So if it doesn't have a mitochondria, what does that mean? Can't make its own energy. So again, when they discovered all these things, they really realized that this organism could not do anything. It had to have a host. So it has genetic material, and that genetic material is either DNA or RNA. There are no viruses that have DNA and RNA together. So it's one or the other. It's either linear or circular. Sometimes they're segmented, so it could be linear with 2 or 3 different pieces. It could be double-stranded, it can be single-stranded. So you can see the genetic material can be a whole wide range of things. And that gives us a very difficult time to deal with viruses because they are so varied. They only can replicate in a host, and what it does when it gets inside the host, it then takes over the metabolism of the host. And that's one of the reasons that the host will often die, is the host needs to do housekeeping, needs to put certain molecules on its surface so it's recognized by other cells, so it's not killed because it's not a complete cell. There's lots of reasons why a cell needs to have its own genetic metabolism to be able to maintain itself. And when that's commandeered by a virus, then sometimes that cell will not be able to survive. And viruses may or may not integrate into the genetic material. So you can imagine if it integrates into the genetic material, that means it's there forever. So HIV, that's one of the dangerous things about HIV. When it infects its host, it integrates into the genetic material. So therefore, it is always going to be with the host. You cannot ever get it out. And unfortunately, the genetic material that it gets into is the immune system. So there are special molecules in the immune system that have these receptors on it that the virus can get in. So it gets into those, and once it does, then whenever your immune system is activated, a virus is activated. So it's really a catch-22. So it's such a very dangerous disease. And we'll talk about it at the very end. Okay, so now we know viruses need to commandeer a host in order to survive, and we know kind of what the shapes look like. Part of these shapes are made up of tiny proteins. I remember you said, I told you that it's only protein coats, and that protein coat will surround a bacteria, or the genetic material of that organism. So the capsid is the name of the protein, and this is the protein coat. So right here is our protein coat. And the capsomer is a single coat protein, so each of these little ones are a capsomer, and then that makes up the entire capsid. And then you have a nucleocapsid, so these are all terms that you'll need to know, or maybe even identify them on a drawing. So this nucleocapsid, here is a capsid with all the capsomers on it. And inside, this is a cutaway, and you can see inside it's housing the genetic material. And when it's in like this, you have the capsid, capsomer, and the genetic material. It's called a nucleocapsid. Okay? So this is about the barest form of a virus that we can have. Then we can get a little bit fancier after that. We can put some membrane around it. We can put some spike proteins on it, etc. So this is just kind of the bare bones for a virus. And a naked virus, this is what this would be called because there's no envelope around it. This is the same virus with an envelope around it, or the same structure of a virus, not the same virus. So it has the nucleocapsid, has the genetic material, and now this one has a membrane around it. If it has a membrane around it, that means it must be infecting a cell that it can bud out of the cell, taking some of the cell's membrane with it. And this gives it an advantage because when antibiotics or something are treated for other things. Antibiotics don't take care of viruses, but when antibiotics or any other kind of molecule in the cell is looking for a pathogen, it sees this membrane and it thinks it's self, because that membrane is the self of whatever the virus gets into. So viruses are surrounded by a membrane, and we call those the envelope ones, and this is the phospholipid membrane of an organism that they were released from. And now a virion is another term, and this is a virus that is outside the cell. So when it's inside the cell, if it is a eukaryotic cell, we call it a virus. If it is a prokaryotic cell, if it's a bacterial virus, we call it a bacteriophage. But either of those, when they're out of the organism, it's called a virion. Okay? It is an infectious particle, but it's just outside the host, whatever that host may be. Okay? And again, now we should maybe identify the shapes. So these are called helical. You can see, and they really look helical, so they're pretty easy to identify. This one is called icosahedral, and this portion right here is what they're identifying. So it's a 20-sided virus, and it's all put together very, very fast. You can imagine viruses have to assemble themselves within a dividing bacteria. A bacteria divides every 20 minutes, so in that period of time, the virus has to get all its proteins and everything done also. So it has a lot of repetition in it, so that it can duplicate itself extremely quickly. All right, so this is an icosahedral virus. They have all this kind of head group like this. And you don't need, at this point in time, you don't need to know any particular names of viral particles. You do need to know what the shapes are, but you don't know that this is what tobacco mosaic virus looks like, or rabies virus. That'll all come much later in the class. Okay, so icosahedral, and now you can see we're combining, so these are called complex viruses. So you're combining two different shapes. Okay, and again, now this is going to be the T4 shape, and this is called for a bacteriophage, and this is going to be a lambda phage. So two different, and bacteriophage and phage are basically the same nomenclature. And then this is the smallpox. It has a totally different shape of any of the rest of these, and there are many other kind of shapes. We just call them complex. So if we compare them, you can see that this one with the helical and then the icosahedral head group, this is a bacteriophage, and then this is like an influenza virus. And influenza viruses, every virus is going to have a unique cell type that they infect. And so influenza is going to have these receptors on the surface that is going to be able to bind onto their target membrane. So whatever target membrane that they have, that they target, this is the cell on the outside, or the structure on the outside receptor that will actually fit into that target. This is really what their icosahedral head looks like inside, and then this is for a influenza virus, they have several different segments. And this is why when you hear about the flu, and maybe the avian flu and the pig flu have combined together, what happens is two different viruses get into an organism, and those viruses, as they're duplicating genetic material, and they have all kinds of segments, a segment of a pig can get into one that's a segment of a bird, and then that makes a new virus. And if that is located maybe in proximity to humans, maybe it's in a pig pen, and it's a duck, and the duck is near the pig pen, and then the farmer goes in the pig pen and harvests or does whatever, then that is where that particle can start to infect a human. When it replicates in a human, now it's also getting human genetic material for the virus. And so what happens then, these viruses can jump from bird to pig to human, etc. So those are the ones that get to be very dangerous. And it doesn't happen very often because it takes a very unique combination. But pig and human are very similar, and bird can easily now infect pig. So it's only a matter of time. So anyway, these are just to show you the two different shapes of what viruses look like. One's a bacteriophage and one's a regular virus. Okay, so here's our first eye clicker. So which of the following is not a part of a virus? Okay? Not a part of all viruses. Okay, so people that have C, a capsid is in every single one that we saw, there was a capsid. But there is something that isn't in every one that we saw. So what would be an alternate? Yes. So it would be B. So there are envelope viruses and there are viruses that are not enveloped. Okay, but they all have to have a protein coat around them in some way, and those are all capsids. Okay, so capsimeres will make up the capsid form. Okay, and they all obviously have to have genetic material. Okay, so how about this one? So this is the one in red that we're talking about. What would that be called? So we're actually only looking at just the head group. So the morphology of the bacteriophage in red would be classified, the head group. So I think the whole virus, that maybe it's confusing, there's a couple terms on here. One would be the whole virus itself, but we're looking at just the head group. So Andrew, do you want to change your thing if you're thinking of just the head group? Because you're right, it's a complex virus itself, the whole thing, because it has a couple different parts, but the head group is one particular part. This is helical, and what is this called? Icosahedral, right. So it is a complex. This is the helical part right here, and then this is the head group, and that would be considered icosahedral. Okay, does that make sense for each part of it? Okay. All right, this is way more than you need to see on one side. It's very confusing. There's nothing on here that you're going to be asked to name now, but there are lots of these that we're going to talk about later on. We're going to talk about different viruses and different diseases that happen. That's almost at the very end of the class when they talk about different diseases. But I want you to look up here. We have DNA viruses, we have RNA viruses, and you can see for the DNA viruses, and this is just a very, very, very small list. So these DNA viruses, you have double-stranded ones, you have single-stranded. Then for the RNA viruses, we have a double-stranded, but we have a positive single-strand, and then we have negative single-strand. And then we have these retroviridase, and these are the ones that are reverse transcriptase. So going from RNA to DNA, just like HIV is. So a very small portion of the viruses that we have are listed here, but we do need to understand when these infect, how does the cell actually, or how does this type of virus commandeer the properties of the cell? So we're going to talk about double-stranded, single-stranded, DNA, and RNA viruses. So the classifications, just like I said, DNA viruses, they're single-stranded or double-stranded DNA, and we saw that. So if you need to replicate that, or if a virus needs to be replicated, it needs to have a DNA polymerase. So the cell that it infects is going to have a DNA polymerase, which most organisms that we know of have DNA for their genetic material. So they do have DNA polymerase. If this happens to be a virus that's going to infect a eukaryotic cell, it has to get into the nucleus, because that's where the DNA polymerase is. So if it's a DNA virus, there's a lot of things it's going to have to do to start replicating its own genetic material. So we'll talk about the steps that has to happen. And then for RNA viruses, you saw a list that was single-stranded and there were double-stranded. And in order to replicate this, we're going to have to use a RNA-dependent RNA polymerase. Okay, and so our cells, eukaryotic cells, don't have those, neither do bacterial cells. So we don't, in either prokaryotes or eukaryotes, we don't replicate RNA. There's nothing in the cell that would need to be replicated. The only RNA that's generally seen in the cell is the mRNA that comes from the DNA in order to have the ribosome in the cytosol make a protein. That's the only RNA that's seen. So that RNA is never replicated. So now we have a virus would have a problem because it needs to replicate its genetic material. And so it has both RNA and single-stranded and double-stranded RNA. So we need to understand how that's going to be done. And then I think you probably know that mRNA is considered as a positive strand of RNA. So there's always a negative and a positive strand, and the positive strand is what is made from the DNA by the DNA-dependent RNA polymerase that comes out of the nucleus or is in the cytosol for bacteria to be read by or translated by the ribosome. So because we don't have the ability to do like negative single strands to be translated, we're going to have to take the negative single strand and turn it into a positive single strand. Okay, so that's going to happen. And then retroviruses, they also come in as a positive strand, but they don't want to be RNA. They need to package up themselves and go into the nucleus. So you can't get in the nucleus if you are RNA. That means you have to switch that RNA into DNA, and that's what a reverse transcriptase does. So these are the type of things we want to talk about on the next couple slides. And it's there's a lot to it, so we'll probably try to go really slow. And then you have to let me know what you get and what you don't get. So this idea here, we're going to talk about HIV only at the very end of the lecture, but I want to bring it in kind of often so you get used to what's happening with HIV. So HIV again is a eukaryotic disease. It integrates into the genome, and when these viruses come in, the integration into the genome is what is so bad. It is in with the T cells, and in your body, in your immune system, the T cell is the regulator of your immune system. So the cell that this HIV gets into, actually infects, is the T cell. And so probably if you've known or if you've heard people talk about HIV, they always talk about the T cell count. When the T cell count is higher than the viral count, the person is doing well. But when the viral count starts getting higher than the T cell count, what's happening is the T cells are infecting more and more, the virus is infecting more and more T cells, and it's making it so the person cannot fight the disease any longer. So anyway, we'll talk about that a little bit more, but this is why this is such bad news. This will come in as an RNA, go to a DNA, integrate into the genome. So this is kind of a review for most people that have had biology. I don't know how much of it is reviewed for you, but we should go through each of these so it makes sense. But just the word replication means that you are replicating exactly the same thing. So we have to replicate our DNA to divide. We have to replicate, viruses have to replicate their RNA. So a DNA replication is going to be by a polymerase. You're just going to polymerize a new strand that looks just like the old strand. So a DNA polymerase for a DNA virus is going to just duplicate the strand. So this is just for the organism to start multiplying and dividing. It has nothing to do with making mRNA, etc. All right, so a DNA polymerase, that's what it does. Then if we want to take any genes off that DNA, we need to have something remove some of the genetic material from that DNA and then take it out into the cytosol, and I'm talking about a eukaryote now. Take it out of the nucleus into the cytosol so that the ribosome can make a protein. So we're going to use an RNA or a DNA-dependent RNA polymerase to transcribe the DNA into RNA, and it's going to be mRNA. So a DNA-dependent RNA polymerase is going to transcribe the DNA of the gene that we want into mRNA. Now the mRNA is a positive sense mRNA. That's what it transcribes. So messenger RNA is a positive sense RNA. This is something recognized by the ribosome. So going down this column, so now we have our ribosome, and the ribosome is going to be reading, translating the mRNA into the protein. So this is just done on the ribosome. This is called translation. There's no enzyme. This is just a ribosome that is going through this motion. And ribosome is not an enzyme. It's a whole bunch of proteins. Okay, so now that we've done this, we've got our protein made, and this was just a big structure of the same thing here. So now we have our protein made and we're done. If we're thinking about, and this is, let's say, just a human replicating, but if we're thinking about a virus wants to go through some of these steps, but the proteins it wants to make are its capsids. So it wants to make a whole bunch of more capsimeres, maybe it wants to make some spike proteins, maybe it wants to make other things that it needs. So it's going to have to go through this. So let's go over on this side and say this is a positive strand RNA. So this is a positive strand RNA virus. It gets into the cell. The first thing that's going to be seen is the ribosome. The ribosome is going, oh, I don't know where this mRNA came from, but I know exactly what to do with it. I am supposed to take whatever's on this mRNA, and I'm supposed to make proteins. So the virus goes, perfect, that's what it wants. It commandeers that ribosome, and the ribosome starts making more protein. So it makes any protein that is in the mRNA strand. That could mean that if the virus needs to make more of itself, we have nothing in eukaryotes or prokaryotes that can duplicate RNA. That means on this strand, and let's say now this came in from the virus, it has a protein called an RNA-dependent RNA polymerase. And that allows it then to take a single strand, a positive single strand, and copy it into a negative strand. It needs a negative strand because it needs to make more positive strand to package into its capsid head group. So it needs this negative strand, which is an RNA-dependent RNA polymerase, that is made from this first strand that came in. The ribosome makes it. It's a protein, just like a capsid protein. That RNA-dependent RNA polymerase then will make a negative strand. It will also take the negative strand and make a positive strand. And we want the positive strand because the positive strand is what we have to package into the head group. It comes in as a positive strand, and it needs to leave as positive strand because that's the kind of virus it is. But to get there, it has to get a negative strand as a template. Does that make sense? So the positive strand RNA virus will bring its DNA inside, and when it is, the ribosome will make any protein that is encoded on this particular strand. One of the things that the virus will do is will encode an RNA-dependent RNA polymerase protein on that strand. When all the capsids and everything else that's made from this mRNA strand, also an RNA-dependent RNA polymerase will be made. It will then take this strand and make a negative strand. And then it will take the negative strand and make a whole bunch more positive strand. And then once all the proteins are gathered together, then for the capsid being made, then the positive strands of RNA will package with it, and the virus will be whole again. It will assemble. Okay? So that's kind of an easy one. That's what happens when you're a positive strand RNA. RNA virus that's a positive strand. So we saw on the list that there were negative strand of RNA viruses. So they are negative single strand. Yes?
So just in this beginning process, it takes from the host's DNA and turns it positive mRNA, positive RNA.
So ask your question again from the beginning.
Does it take from the host's DNA to turn it into a positive RNA?
Oh, okay. So this is a little bit confusing. This side is just how I reference what happens in a normal cell.
Okay.
So now we're saying, we'll just take this side. We'll eliminate that side. So we have just this side over here, and a positive sense comes in from the virus. So from there, then with the ribosome, that is the host ribosome, but the ribosome then leaves the host, not leaves it, but ignores the host, and does whatever the virus wants. And the virus tells on its positive sense RNA, it tells the ribosome what it needs. And it says, first of all, I need an RNA-dependent RNA polymerase so that I can make my other sense of RNA. And then I need you to also do the proteins for my capsid and anything else it has, my spike proteins, whatever the virus needs. So yes, so it doesn't get anything from the host, but it does take the host ribosome to do this.
Okay.
Right, so now if you're a negative strand, and you're a negative strand that you come in, so basically you come in as a negative strand, the ribosome goes, what is that thing? I don't know. I mean, I don't recognize it, so I can't use it. So that means that if this encodes for a RNA-dependent RNA polymerase, it's not ever going to get read. So it has to bring one in. So it's not shown here, but there'll be a little protein that when it comes in is attached to it. And that protein is going to start right away taking this negative strand and making it into a positive strand. And then the ribosome goes, I recognize this thing now. So then it will actually have the ribosome make all the co-proteins, etc. But then this RNA-dependent RNA polymerase that came in with the negative strand of RNA, it's usually called just the word replicase. So a lot of textbooks will say replicase comes in attached to the negative strand so that it can immediately start making a positive strand, which will be recognized by the host ribosome. So it is an RNA-dependent RNA polymerase. There's a whole list of what things are included in that nomenclature. So we'll just always stick to RNA-dependent RNA polymerase so you don't have to memorize several things. But I think maybe a couple of slides we'll call it replicase also. So the RNA-dependent RNA polymerase will come in. It makes a new strand. It's now a positive strand. The ribosome can now make lots of proteins from that are needed. And then also the RNA-dependent RNA polymerase can come back here and make more negative strands. So if it comes in as a negative strand, it must be packaged as a negative strand to stay the same kind of virus. So more of the negative strand will be made, and all the capsid proteins, spike proteins, whatever the virus needs will be made. Okay, makes sense? But it can't be made until the replicase that comes in, or the RNA-dependent RNA polymerase that comes in, gets the positive strand of RNA made. So a little bit more complicated for this virus, but it definitely works. Okay? And you can see how much work the host ribosome is now doing when a virus infects. And when a virus infects, it's generally not one virus. It's many viruses. And so therefore, the ribosome for the host is so busy making all the proteins for the virus that often the host will start to die. Okay, it just doesn't make any housekeeping genes. It doesn't make anything that the cell needs. Yes?
And then quick question for when we have it referenced as like positive SS RNA, is that for positive sense? Is that what that
A positive single strand.
single strand?
Yes. So the sense of it is this plus or minus, and the SS means single strand.
Okay.
Yeah, there are like double strand, there's a whole bunch of different things, and we're just going to kind of go through the complications of a few of them, just so you get a feel for what viruses do. Okay, so I think those are the only ones that I wanted to go through. Let me make sure there's... If I've forgotten one, we'll come back and we'll talk about that. Oh, I guess DNA, that's probably a little easier to understand. If a DNA virus comes in, it kind of follows this trend. It doesn't replicate itself, although it will have to replicate itself in order to put it into the packaging. But it would happen if you're a eukaryote, not a prokaryote, because a prokaryote is not, it has a nucleoid, but it isn't separate from its cytoplasm. So when the virus comes in, the nuclear material for a DNA virus is right there. They don't have to go into the nucleus. For a eukaryote, it has to go into the nucleus in order to use the DNA polymerase in order to polymerize that DNA molecule. Okay, but the rest of it is the same. From that DNA molecule, it will be transcribed by DNA-dependent RNA polymerase in order to make an mRNA. That mRNA then will be read by the ribosomes. Okay. All right. So what about this question? It says, what do ribosomes do? So this is where nomenclature is really important. Okay, anybody else have any more? Isman, do you have an idea? Yeah. Okay, so, or maybe you were just flashing that. Okay. Okay, so yes, and I mentioned because I have a couple of questions coming up, and if they are really difficult for you, you should really go back through the previous slide I did and go through it over and over again. So this one, yes, you should be able to distinguish between replicate and translate and transcribe. These are all things to kind of test, if you've got the right concept. But yes, mRNA is translated into protein. All right, so here's one that's a little bit more difficult. So which enzyme is paired with the incorrect function? So enzymes are always ending in A's. So we've got an A's up here, we have an A's right there, we have an A's right there, we have an A's right there. So these are 4 enzymes. Okay, so it looks like most people got the answer correct. It is C. So the RNA-dependent DNA polymerase replicates RNA to double strands. So an RNA-dependent means it is depending on RNA to double strand, but that's going in the wrong direction. We don't do that. Okay, so this would be the wrong one. Okay, make sense? So again, it's just getting the replication or the polymerase or the translation, all of those terms correct. So you'll see multiple choice kind of questions like that, or maybe a short answer to say, how would this happen if this got into the cell? So you'd have to have a kind of a short answer on that. Okay, so this is kind of a review of the previous slide, but we see it in a more realistic context. So here's a single-stranded RNA coming in for a virus, and so the virus brings its single strand into the cell, and then this is going to have to be, right away it can be translated by the ribosomes, so you can see it's translating and it's making all the proteins. Those proteins then can be assembled down here, but the other thing that has to happen is this has to go into negative strand. So we have to have an RNA-dependent RNA polymerase in order to make a negative strand, and then this will be the template that's also going to be packaged in with the capsid, etc. So just like a little linear progression of what happens. And then you just fill in the blanks. So you could actually see something, one of these panels or something like that, where you need to fill in a little bit more detail on each of the scenarios. Like going from here to here, what actually is going from there to there on this particular side? So what thing is missing? If we're going from here to here on this side, what's missing there? The word translation's okay, but something's missing to do it.
Ribosome.
Ribosome, yeah. So maybe a figure like this where you're filling in some of the terms to say what happened. Okay, and then the next one, this is a negative single strand, so we have negative single strand come in, and of course we've got, it has to bring in something. Generally the textbook calls it just the replicase, but that's an RNA-dependent RNA polymerase. It just has to make this into a positive strand. And then, of course, we're almost going like this side. We have to translate those. We have to then make a new strand to package in. We always have to have the same strand that we came in. We have to have that to package in when we're assembling. And this one, we're not going to talk about this one. This is just, I didn't talk about it, and it's just more that you would have to be thinking about. So these two mainly are the focal point. Okay. That makes sense? All right. Here's a table. They have some verbiage on here I think that is very useful. Remember the double-stranded RNA here, we didn't talk about this, but all of this up here is stuff that we talked about, so this table would be good for a quick review to kind of read it just to get some verbiage down. So I'm not going to dwell on that one, but that one's also in the text. All right, so now that we know that viruses can actually infect organisms, how does it do it? What is the process for doing that? So now it's getting darker outside. I can see the slides look so much better. It was like getting really light. But anyway, okay, so viruses, they have to be able to get into their host, and that means they have to have something on their surface that matches with a receptor on the surface on the host, which is really an alarming idea if you think about it, because why would our cells ever have a receptor to accept a virus? I mean, genetically, why would we waste our time making those receptors when we never expect to have a virus infect us? Why would we make it so easy? So anybody know on COVID what receptor that the COVID virus was binding to get into cells? So there's a receptor called the angiotensin receptor. So it has a lot to do with heart rate and hypertension, etc. So that's a virus has nothing to do with that, and yet it has mimicked the ability to be able to bind to that receptor. So it's very, very specific. HIV, it binds to a receptor, CD4 receptor on a T cell, one of our immune system cells. And that's the T cell never planned to have it being bound by something like a virus. And so all of the receptors that are used by viruses are not for the virus, it is for something else. The virus is just mutated and mutated and mutated into a way that all of a sudden it now matches a specific receptor in a host cell. Okay? And you know, viruses, they have a very poor a replication system, and that replication gives them mutations over and over and over again. And it's just unlikely that one time over years of replicating that virus is going to match a receptor that's in a host. And once it does, then that locks into that one, and that virus can multiply with that exact genome and bind that host. So the host range that viruses have, they have to be host specific, first of all, and then they have to be tissue specific. When a virus gets in, it's the likelihood of it getting to the right tissue is usually pretty far-fetched. So many viruses we are not even affected with. They may enter the body, but they never get to the right target. And you see, if it's the lungs, let's say the target is the lungs, and you swallow a virus, it's never going to make it to the lungs. If you inhale the virus, it's going to make it to the lungs. But if you have something that's supposed to infect your stomach, and you inhale it, it's never going to get to the stomach. So it's those kind of things that it's almost remarkable sometimes that a virus can even infect a person, because there's a lot of ways it could go wrong. Viruses have a host range, number one, and then they have to have a tissue specificity. That means they have to get to the tissue that they're going to infect. Viruses cannot just attach to any specific receptor on any tissue. It has to be a very specific receptor, and those are usually on specific target cells. So if it is an envelope virus or a capsid, it's going to have kind of a different structure. So here's one with an envelope, and if you have an envelope, you can put spike proteins in the outside of the envelope so that they are available to be able to bind as for your target cell. So here's one that's an envelope. Here are the target receptors that it wants to get into, and so whatever we'll call them spikes, the spike proteins will actually be able to match up, and then it will trigger an endocytosis. Here's one that it's just using the spike kind of things that are on the capsid itself. It can have these long spike-looking things, and those will actually fit into receptors also. So they will have the same effect. They have very host-specific receptors. So they have to be tissue, cell and tissue tropism just means what they love to or what they're bound to get into. So tropism is just the very specific area. And there's receptor specific, so I think I went into that. So a host, when it invades, sometimes they don't have one receptor, they have several different receptors to cause them to be able to get in. Here's a case, we'll look at both of these, HIV and SARS. So HIV, it needs CD4, and I mentioned that a few times, so that's something that you should remember. CD4 is the receptor on our macrophages, on our T cells, two of the primary molecules of your immune system. And HIV can bind to that receptor and get into those cells. So CD4 is a receptor, and then it has a co-receptor. It's called CCR5. You don't need to know that. And CXCR4, you don't need to know that. But you do need to know that CD4 has a couple of co-receptors that allows the HIV to enter into cells. And this was very interesting when they did research on HIV at the very beginning. They noticed that sex workers in Africa had many, many partners, and they never got HIV, and the partners all had HIV. And they couldn't figure it out, so they finally did some genetic sequencing on these individuals, and they found that they did have a CD4 receptor, so they were susceptible to being to have the virus bind, but they didn't have CR5 or CXCR4. They didn't have the co-receptors. So it made it less likely for that individual to have the virus actually be able to attach long enough to get into cells. Because in order for a virus to get in, it has to attach long enough to be able to actually get into the cell. So if it falls off, then it's not going to be infected. So anyway, these are very important. They're not essential, but they're very important. And then SARS, also ACE2, which is the angiotensin one, and a protease enzyme also. So I'll just show you those briefly. This is just for your information. This is nothing you would have to draw or do. And do you need me to turn the lights on? Can you still see over in the corner? Okay. So if you can't, let me know and I'll try some lights. All right, so the next slide, we can skip this. We don't have to go through that little thing. So this is actually HIV. Here would be the virus up here, and then here's the viral membrane, and then this is a receptor that is sticking down from the membrane, and this is the one that wants to actually get to the CD4 right here. This is CD4. So this receptor is mimicked here in this green color. So this is what it looks like when it gets near the membrane. CD4 and the virus recognize each other. So this starts to hold the virus close, and then another fusion peptide actually comes in and kind of fuses to these areas right here. So it kind of starts pulling the virus closer to the membrane to have the two membranes, one from the virus and one from the host cell. It fuses those two membranes together. And here you can see the fusion is getting closer and closer together. So some of those fusion proteins and stuff are this CXCR4 and CCR5. Again, you don't have to know the names, but you just know that they are important in getting the virus and the host close enough in order to be able to infect. Okay, and this is probably why the sex workers not having those were infected a lot less. So this is just for your own information. This is what happens when HIV infects. And these are proteins. These are just stylized views of what a protein would look like on the host cell. And then this is just SARS. We're kind of past SARS now, but it's always good to understand how it actually infects. So here are receptors that are on the SARS envelope virus. And you can see these are the ACE2 receptors on a host cell. So as it comes in, it will bind to these host receptors. But then it's not really good to have these. These aren't strong enough. So what happens, there's a transmembrane protease that is going to cleave these off. And you can see these are cleaved off. And it leaves these other portions that are able to bind to this TMPRSS2, whatever that means, probably transmembrane something or another protease. And what this is going to do is it's going to pull it in tighter. So as it pulls it in tighter, they kind of withdraw into the cell and they pull this in tighter. And eventually the envelope on the SARS and the membrane of the cell fuse together. And then this is allowed to get into the cell. So viruses, it's almost remarkable sometimes that they actually make it into the cell. There are so many things that have to work exactly right. But this is how viruses infect. So now that the virus is in contact with the membrane, they are able to come in and dump out all their contents, which is really their capsids and their genetic material. And from there, we know what happens. Okay. So just for information, and that's all, this is not anything that you have to know except the top one. So these are just some viruses. Some of these we'll talk about later, ones that cause cancer. And then these are the cell types that they actually infect. So CD4 positive T lymphocytes, that's the T cells that regulate your immune system. And then macrophages, the two ones that have the CD4 receptors on them. Okay, and then the rest of these, you can see all of them have different places that they are infective in hosts. And these are just a very, very partial list. So it's each virus will have something very specific that they're going to be binding to. Okay, so the only one I really want you to know and be able to recall is HIV, only because we are going to talk about HIV a couple times in the class. All right, so what about this one? It says, which of the following is true? Oh, you might not be able to answer this. I didn't cover some of this. I moved a slide. We'll see what you say, and then we'll... I think some of it you can do. So you're saying Yes, yeah. So everybody that said C is right. B, bacteriophage can infect humans. They can't. Thank goodness they cannot. I didn't talk about how viruses are grown yet. I moved that slide to the very end of these, so that would have been fair to ask. But again, a kind of a question that you will see on the upcoming exam. So here's exam 2, 5, 6, 9, 7, 10, and 13, which is this chapter, and that's 416. All right. All right, so now how does this whole thing work? I think I'll keep talking for just a little bit, and then we will take a small little break, and then we'll end up pretty soon. There's a standard way that organisms infect, whether it is a bacteriophage or whether it is a virus that is for humans. So these are some of the steps. The virus has got to encounter and attach to its host. So again, like I was saying, if you swallow something and it is meant for the lungs, that virus is never going to make it to the lungs. So it has to be in a pathway that gets to its target, and then it has to be able to find its target. You know, there's lots going on in the body. It has to actually get to the target, has to bind to the right receptors on that target, which, you know yourself, if you're drinking a drink and you have to put the cap back on, you have to almost look where the place is you put the cap, because it's not, you can't just reach and do it. You usually have to look and target it. So how a bacteria, or how a virus and its host receptor actually come together is pretty amazing. And then it has to invade, so it has to actually get in, and then it has to hijack all the metabolism of the system. So there has to be, the ribosome has to be available, the cell has to be healthy, has to be able to make all the stuff available for that virus. And then it has to assemble in the host, and then it has to escape the host. So the host usually has a membrane around the cell, so it has to get out of that membrane and get off into something else. All not being detected by the immune system. So these are the things that will happen. You'll see these 5 steps over and over again. I've already mentioned this, but we have 2 kinds of viruses. Bacteria viruses are called bacteriophage or just phage, and animal viruses are viruses. So, and in humans, we're animal, and so we have viruses. I slip up a lot, and I call everything a virus. Okay, but I never slip up and call everything a bacteriophage. But I do find myself saying the word virus when I mean bacteriophage. But in writing it, you'll be doing slow enough and stuff, you wouldn't make a mistake between the 2. All right, so we're going to go through the bacteriophage first. So again, we already know what kind this is. This is a T4. This is what we're going to be dealing with in lab. The first step is the attachment. It has to absorb onto the surface. So a lot of times instead of attachment, you'll see absorption. And then the tail fibers, you can see on this particular type and style of bacteriophage, it has tail fibers. For the other one that we saw this helical thing like this. This doesn't have them, and this is called lambda. So this is a lambdaphage, and it doesn't have anything. It just on the bottom it would have attachment places, but it's just helical like this. And those are the 2 kinds of bacteriophage that we'll talk about. So this is a lambda, and then this is the T4, and that's what we'll do in lab. So it's got to attach, and this attaches with its tail fibers, and this is just going to attach with proteins that are on the end of the helical structure. And they have to match with a receptor. And then it has to penetrate in. So most of as this is a bacteriophage, we're talking about bacteria. So most of the bacteria have what boundaries on them? Let's say it's a gram-positive bacteria. What boundary does it have for this phage to get through? Thick peptidoglycan. So how is the genetic material going to get through? It's going to secrete this thing called lysozyme. And this zyme here means it's an enzyme. And this lysozyme is going to secrete a material that will break up. It will break the bonds. Remember, peptidoglycan has N-acetylglucosamine and N-acetylmuramic acid. They're sugars that are bound together that kind of keep the peptidoglycan partially together. And so this lysozyme will actually dissolve part of that cell wall by breaking the glycosidic bonds. Okay, so when it breaks those glycosidic bonds, it weakens that cell wall, and then the genetic material from the bacteriophage will be able to penetrate that. So it really doesn't, it shows it a little bit over here. What happens is there's this sheath, kind of like a hypodermic needle coming out of the bottom of the bacteria, and also it secretes the lysozyme. And what it does is it squats down, for I guess a better word. It gets really close, and then it injects its DNA inside. Once it's got the lysozyme secreted and the cell wall starts breaking apart, then it injects its genetic material into the cell. Okay, once that happens, now the first step is going to be what? So now we've got the genetic material in there. Let's say it's a positive strand RNA. What's going to happen almost immediately? Ribosome is going to be right there. It's going to start replicating. So the biosynthesis portion starts happening right away. Whatever kind of a bacteriophage it is, it's going to start the proteins are going to start to be made, new genetic material is going to be made, and it will start, this is called the biosynthesis part. The new phage components are all being made. Genetics, genome, and all its capsid and receptors, whatever it needs. Okay, so that's the biosynthesis section. And then it starts maturing. That means we have to wrap it all up to start making a new phage, bacteriophage. That means we need to assemble all those parts together, get the genetic material inside the capsid. You can imagine how hard that can be. If you're making a capsid and it has so many little capsomeres and they're putting them all together to make the structure, and then before you close up the last little hole, you have to say, wait a minute, we don't have our DNA yet, and you get the DNA packaged in there. It's amazing that even a bacteriophage would ever be able to assemble itself, but it does. And then once this is called the maturation, it's going to put all those pieces together, and then it's going to be released. So when it's released, there's two different ways it can do it. It can go to the boundaries of the cell and just burst out. It can secrete some more of that lysozyme and break down some of the barrier and then just burst out of the cell, and that of course kills the cell. Or it can, has another different avenue it can take when it first comes in, is it can be a lysogenic phage where it actually starts integrating into the host first before doing anything else, and then it just multiplies with the host every time. And remember, most bacteria multiply about every 20 minutes, so that means that that virus embedded in the host of the bacteria are going to multiply with the host every 20 minutes. So in first 20 minutes, you got 2 more cells, and then 4, and then 8, and then 16, and after a certain period of time, you have 100,000, and then you have 200,000, and then 400,000. And all along, you also have that many number new viruses that are embedded in that bacteria. So really what you're doing is you're just, you're propagating the bacteria, but leaving it inside a bacteria. So anytime the virus wants to get out of all those propagated bacteria, it just has to bust out, and now it's in a new environment, maybe a lot more bacteria nearby. Okay, so these are the steps, and so now I want to show you this whole process. But maybe before I do, we should take a quick break. And then let me, because I know you'd like to know how much longer. So let's just go through this really quick, and you kind of see the number of slides that we have, and maybe not. Maybe I can't do this really quick. It doesn't like the quick motion. Oops, don't look at those. So this is about cancer. This is a stopping point, but I think this is important. And then this last part is also a stopping point. We need to know this. This is also a stopping point where we have the virions and we have the prions. So we can eliminate those and just, you can glance at those slides and read the text. But or we can go through to the end. So we do have a couple stopping points in there. All right, so not so much more. So let's go ahead and take a quick break, maybe 10 minutes. Okay. Let me just kind of write what our times are up here. Oh, here it is. So, looks like it's 7, maybe 12, so maybe 7:22. Okay. Now watch, nobody moves.
I'm a move.
I think it's like halfway through.
Well, I flashed you the slides. Did you see how many?
Yeah, well, I have like 20, but I meant like in terms of like time. Will the second half take just as long as the first?
I think so, yeah. And I don't know how many slides are left, so I think it usually is like 2 to 3 minutes per slide, so, I mean, it kind of depends. Oh, I ended up with the sky was. Yeah, there's not quite as many clickers, so that makes it faster also. Okay, so the board isn't very light, but there's a thing on exam 2, exam 3, and the final exam, so you should know that. What is this? This is interesting. And nobody needed lab report printouts? That's awesome. Also, I think I told everyone that I always bring the textbook, so if you need to take any pictures from it. Also, the textbook is on reserve at the library, so if anybody needs that. I'm hoping maybe sometime in the latter part of the weekend I'll have your study guide for this done, and of course it always references certain things in the book, so if you need to, you can look at the book at the library. I think they said there's 2 on reserve. Yeah. I don't know how available, when I really think about it, having only 2 books on reserve, since all classes are, all the ends, all the exams and stuff and quizzes are all scheduled at the same time, because we all have the same syllabus. But they told me they're never used. Nobody usually uses them, so, because I said, "You think we should put some more?" And they go, "Oh, no, they've never checked out." So I'm like, okay. So I suppose if it got so that there was a lot of people checking it out, maybe they would put more on for sure.
The Pearson one's pretty good too. The Pearson one, online textbook was, has some like AI thing, where it could be like summaries of different questions, chapters. Yeah, that one's pretty good. It's good for study.
I just wondered how long the quizzes and exams are. My brain will not be able to take the overload of the textbook, you know what I'm saying?
Yeah.
Like if I can know anything over
Yeah.
study or something.
Well, there's a lot of white noise, that's why I highlight, but I want something like that.
That's why I do the summarizing things too, like different like sections of the chapter, just like giving the key points and like summarize it up to a smaller thing.
Are you talking about the textbook? Because they really have good little sections in there where.
I'm talking about the Pearson one, the online textbook.
Uh-huh.
It has Pearson added a function where they have some AI thing that is able to summarize different portions of the chapter for you, so you're not reading the entire.
Yeah.
Because there's a lot of information that we have to have for the next exam, so it helps with summarizing certain parts, so you're not just reading constantly and not retaining information. You know.
Yeah, the text itself also has sections where they summarize everything, and usually after I make some questions, I kind of read that and I go, oh, there's the question right there, answered. So usually I pick what they also summarize, but not always.
They have questions at the end of each chapter as well. Do you ever take any information from those?
Yeah, when I give you the thing, I'll say, make sure you look at, and then I have like visualizing. I like those. I think there's a couple things in here. I said, make sure you understand this, because there's a visualizing conceptual idea in there. And so then I can just use their figure and then use what they said.
Yeah.
Yeah. So yeah, I use a lot of that.
No. How are you? How did that cook in the car? Can we eat in here?
Pardon?
We can eat in here, right?
I don't know. There's no sign.
Just not in lab?
Oh yeah, in lab I know we cannot, only because of chemicals, but I mean some rooms, like at Fullerton, they actually have big signs all over that says you cannot eat in the room. You would never know it. There's stains all over the floor where people drop stuff, and they always have things. The only thing in my classroom is I say, please don't bring like food that is warm, because then it has an odor. It's like French fries or hamburgers or, yeah.
Yeah, from what I know, I think that way you can. I've been on many classes and they let you eat.
Yeah, I mean, I don't see any sign, usually if they're really, specific about it. It's just that, most of these carpets, you can almost clean up anything that spills on it as long as you clean up pretty quick, so. But they got the rolls of paper towel here. Because I think this is like indoor, outdoor, or something like that, where it probably cleans up pretty good. Since we are a guest here, I don't want to screw anything up. Okay, so I think we have everybody back, do we? Yeah? Are we missing anybody? Okay. Yeah. Oh,
yeah.
so we would get going, but we'll wait just a second. I always have a trouble getting the projector on, and this place, this room has a thing that you turn the projector on with this. So I think if that happens again, I'm going to come over and get this and see if it turns the other one on, because it's always so hard to get it on. Okay, so if everyone is ready, you can eat, and I'll just talk about our viruses. for a bacteriophage instead of viruses. So again, I said you need to know that this particular one is a T4, and this one is a lambda. Okay, there's the two kind of bacteriophages that we will talk about. All right, so again, in here, it means a virulent phage just means a phage that's going to kill. And a virulent phage, we've got two kinds. We have a lytic and a lysogenic. And a lysogenic just takes a little bit of time on the killing. But both of them, their whole goal is to make more phage and then have it bust out of the cell. Okay, so this is really divided into two. And so the first one I'm going to talk about is this stage right here. And then we're going to come back and talk about this stage. This stage, though, is really incorrect in this textbook. I don't know if this is maybe, I'm not sure why they do it this way. I think they made a nondescript phage right here. That phage could be this phage, or it could be a rocket ship looking phage, so that they can do both. But a lytic phage over here, when it comes in, it also cannot be a lysogenic. It is only one or the other. So this one is always going to be lytic, and then this one over here is going to be lysogenic always, and this is the one. This is the one that will be lysogenic. So the lambda phage is, but then when it wants to bust out of the cell, it has to go this route. Okay, so, but that, for quizzes or something like that, I'm not going to ask you to differentiate between the two. So let's start with this. This is the attachment phase. So I kind of numbered them 1, 2, 3, 4, and 5. So here we're attaching. So you can see that the tail fibers, which we're not looking at in here, but somehow it's attaching. And it's using lysozyme. So if you had to discuss a drawing like this, you would need to mention things like lysozyme. It attaches with its tail fibers. It uses lysozyme to be able to break the glycosidic bonds of the peptidoglycan. And it does it whether it's a gram-negative or a gram-positive. All right, so anyway, kind of a discussion is not just attaches. That's really not everything that needs to be said. So here we are, it's attaching. It's going to put its double-stranded DNA inside the cell. And these, I don't know what happened to this thing. That thing should move up a little bit. So it gets its double-stranded DNA inside the host. Here is the host genome. The host now is a bacteria. And once its genetic material is inside, then it's going to go into this route. So you can see it's cyclizing. So this genetic material of the bacteria with the virus, bacteriophage, will actually start to cyclize. And then what will happen is all of this material will be packaged up. So in the meantime, we've made all of the coat proteins, we've made all of the materials that need to be made to assemble our virus, and then we've taken our genetic material and we've duplicated it. It wants to be duplicated in a cyclic form. It's much faster to do that way than if you have to do linear. So it's a cyclic form, it's duplicated, then it's opened up, and now it's packaged into each of the bacteriophage. So that is your assembly and maturation. And so now once you've matured, now you want to actually bust out of the cell, and that's exactly what happens. They lyse the cell. Again, they're going to have to use lysozyme in order to help break open the peptidoglycan to be able to let it out. And so now it kills the cell. Okay, so here are some directions, or some kind of verbiage, but it doesn't say everything. So don't, you just look at this and make sure you're thinking about some of the other words that we included before. So this would be for our T4. So now let's say we have our lambda comes in. So lambda comes in the same way, goes to the same thing, it's going to cyclize. Then the next thing that's going to happen, if this is a lambda phage, is it's going to integrate as a prophage. Prophage just means before it's going to be a phage again. Now it's in the genetic material of the host. So it integrates into the host, and now as the host divides and multiplies over and over and over again, it will take the lysogenic phage, or the prophage that's contained within the genetic material of the bacteria, and it will be multiplying and dividing it also. So we know after about 24 hours you have more than 1 billion bacteria. So this means we have 1 billion new viruses embedded in these bacteria. So if they all bust out at the same time, you would just have a lot of virus. All right, and they stay that way. It could be for days or months, or they don't know what triggers it, but there's usually something that will trigger it. And sometimes that thing that triggers it is another infection with another virus. Because when that happens, if that virus is going to be lysogenic, that means that this virus needs to get out of the genetic material because it will kill the bacteria, and then that means that genetic material will be lost. So it needs to get out and then replicate itself and then leave the cell. So there are various things that trigger this being, instead of being lysogenic, being a prophage and staying in the bacteria, and then actually coming out as a virus again and being assembled and busting out of the cell. They don't know exactly what triggers it. It could be things like density, it could be lots of things, because why bust out if there's not any more bacteria near you that you're going to be able to get in and infect? So there could be lots of cues or something that it understands that can make it the most opportune time to be able to get out of the cell. Okay, so this is bacteriophage replication. So we have lambdaphage, which is lysogenic, and then we have our T4, which is going to be lytic. This is what it looks like. We have entry, so at the very beginning, and it takes as far as minutes, and then all this synthesis and assembly, and then busting out. And you can see about 25 minutes later, the bacteria, especially if it's going to be a lytic phage, the bacteria is ready to infect another cell. So this can happen if you have lots of bacteria cells and lots of virus get out. They have lots of targets to be able to hit. They can kill a lot of bacteria. And what we will see, I don't know if I have a slide right here, no. What we will see is you're going to have a plate on the Tuesday that we come back. You're going to spread a bacteria on this plate. And let's just say that's totally spread with bacteria. And you're going to mix some bacteriophage in with another solution, and it will be infecting on this plate. So it will be all over here, and we won't be able to see it. But by the next day, and I'll just do a little plate right here, by the next day that we look at it, we will have one little spot here that will be like this. And that means that there was a bacteria that was infected by a phage, and that phage busted out of that bacteria. It went to the one next to it, and to the one next to it, and 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 pretty soon you have a big plaque. It won't be that big, but it'll be pretty big. It'll be like one of our discs that we do for our antibiotics. It'll be about that size, depending on how long we leave it, but usually that's about the size they get. Okay, and then that's pure virus. That's all in that clearing area. That's pure virus. So if you wanted to study that virus, you could take a pipette tip, stick it down into that, and you get a little plug of agar with virus in it, and then you could actually start studying that under the microscope, etc., whatever you have, like a scanning electron microscope, our microscope, we look at it. But that's how we study them. Okay, but anyway, this is our the starting thing. We have an entry, synthesis, assembly, and then we bust out. So that's what we're going to see over time when they're all busting out on our plate that we have. So we're just going to put the phage and the bacteria together, plate them, and then see what happens. Okay, so animal is exactly the same way. So I don't need to take a lot of time on this, but we'll talk about each of the same steps. So what you want to do is also look at the two methods that happen. Think about the lysozyme, think about the tail fibers, think about the differences between what we're going to talk about now and what we just talked about for our bacteriophage. So the names of the steps are the same. So we have attachment that are attached to the host. Here we have spike proteins that are going to be attaching to the host, or maybe we can have a spike or kind of things, or whatever we're going to have that are going to recognize the receptors on the host cell. So that's what's going to be attaching. And then the penetration, and think about how these are different when we have a bacteriophage. And then the penetration, the receptor sites vary depending on the people. The virus is usually going to be taken intact. So again, think about what happens. Does the virus go intact, or does the virus fall off and only the genetic material goes inside for bacteriophage? But for animals, the whole nucleocapsid, and that's the capsid, and not the envelope part, but the capsid and the genetic material inside. That's going to actually go inside of the eukaryotic cell. And now once it's inside, it has to uncoat. So here it is going inside, so the whole nucleocapsid is going to come inside. And then, doesn't show, then you would see that that part has to fall apart so that we can get the genetic material out. Yes?
Can you give like an example of how the receptor sites vary from person to person?
Let's say the sex workers. They didn't have the CXCR5 receptors also on their surface. So all of us have different kinds of, we have the same basic receptor, but also sometimes we have sub-receptors. So there could be differences in that way.
Okay.
Yeah, but we have to have a receptor that's exactly going to match with the bacteriophage, the virus, in order for it to infect the cell. So just some people may not have, genetically may not have those receptors on their surface. So that virus, that particular virus would not be able to infect that person. Okay. So again, attachment, penetration, we're in, uncoating. So once this gets in, it's got to uncoat because all of this material has to leave in order for the genetic material to escape. And so now biosynthesis, same type of thing. The genetic material has to be duplicated. All the receptors that are going to go on the outside, everything has to be, whether it's a double strand, single strand, positive, negative, whatever it is, it has to be duplicated in exactly the same way to be packaged up with the capsid proteins. And then this is the maturation. This is where you have to put everything together to make sure the capsid, when it leaves, or the virus itself has everything included with it in order to leave the cell. And then it's going to have to be released. So these kind of viruses for animal cells, they usually just bud out. The animal cell usually takes about 20 hours to 24 hours for a human virus to multiply itself, and then it usually just buds out of the host. And usually not that many bud out at a time, so it's not usually a killing event. But it weakens the host usually enough that if the virus stays within the host and constantly is released, it will actually kill the host. Okay, so it's just more of a measured, and I'll show you a slide on how measured it is. It doesn't bust out all at once. Yes?
At which stage of this process, if any, elicits an immune response from the animal host?
So it's a really good question, and I think in a couple of lectures we will talk about our immune system, which is killer. Your immune system has to be able to see the virus. So the only time that the virus is available to be seen is when it's in the cytosol, or it's in the bloodstream, or it's going to its target. Because once it gets into a cell, it's now gone from view. So your immune system is only exterior. It doesn't get inside a cell and look. It only looks around in a cytosol for your immune cells for its target that it's looking for. And once that target gets in, that's history. Now, the only thing I can say with that is things like cancer. We have our immune system, we have natural killer cells, and those natural killer cells go around and look for common molecules that should be on a cell. And if the common molecule is not there, the natural killer cell kills the cell. And the reason that natural molecule isn't there, that normal one isn't there, is because a virus or something, maybe cancer, has infected a cell, and it's taken over all of the actions of the cell, and the cell does not have time to put that common protein up on its surface. So your immune system, your natural killer cell, says something is wrong, I don't know what it is, but I'm killing the cell. So virus could be infected that cell, cancer could be infected in that cell, keeping the cell from doing the normal things it does, and just from that reason alone, the cell will be killed by a natural killer cell. That's what your immune system will do to help. So, but once it's inside, your B cells, your T cells, those cannot recognize the virus inside. Yeah, but only your natural killer cells do. And cytotoxic T cells too, they also do. Okay, so, answer your question? You good? Okay. All right, so now we're released. The virus is now outside. Once it's outside the cell, yes, it can be recognized by anything in your system. And of course, viruses have lots of clever things. If it buds out of a cell and it has the cell membrane on its surface, you look a lot like a cell. So most immune cells will actually not recognize that virus if it has an envelope of its own cells. They're trained not to kill anything that's a self cell, but eventually, if it does not put all the proper things on the surface, it will eventually be killed. But by that time, it's already into another cell. Okay. All right, so here is the same thing that we looked at before, but this is going to be an attachment with a DNA virus. So you can see here's an enveloped DNA virus coming in. You can see here's its DNA. It's going to fuse with the membrane, so here's the attachment. It has receptors that it can actually attach onto the surface, and now it's going to attach to the cell, and then it's going to penetrate to the cell. So when it does that, the whole nucleocapsid goes in. And then once that nucleocapsid is in, the DNA still has to get out of this. This happens to be a DNA virus. So we need to uncoat, that's what this is called, when we're removing the capsid proteins from the genetic material. And now it goes straight for the nucleus. And of course, there's things that have to help take it there, but just a kind of a shortcut. The DNA then goes into the nucleus. And once the DNA is in the nucleus, we have a couple things that are going to happen. We want to get an mRNA, so we have the DNA-dependent RNA polymerase that is going to make a single strand of mRNA, and this is being transcribed from DNA to RNA, and then we go from RNA and we go outside the nucleus, and when we're outside the nucleus, then the DNA, the mRNA is going to be translated by the ribosomes into all the proteins that are needed by the organism. So again, this slide is a really good slide that you may see only parts of the slide, and you would have to discuss what is happening. Here are some key words that I put so that just make sure that you understand when you're going from here to here, you're transcribing. You need to know what enzyme would be doing that from the ribosomes. You need to know that that is going to be translation or making a protein. Here are all the capsid proteins, and it would be making any other proteins. And here are the spike proteins. The capsid proteins are going to have to go back into the nucleus because this needs to make the capsid that it's going to house the genetic material. Now, sometimes that is going to be done in the Golgi or in other parts of the cell, but we'll just say it's going to be done in the nucleus. So once this is done in the nucleus, this will go to the nuclear membrane where the spike proteins are located that need to go on the exterior. So it will bud out of the nucleus at the point where those spike proteins are. And that means this envelope that is going to be around this particular nucleocapsid is going to be from the nucleus. So that's a nuclear membrane that's going to be surrounding this this nucleocapsid. So then it's going to fuse with the Golgi, and then the Golgi is going to take it, this little yellow membrane is the Golgi, and then this Golgi is going to take it to the surface of the cell. And the Golgi membrane, which is exactly the same membrane as the surface of the cell, it's going to fuse with that, and then it's going to allow this to go through exocytosis and be removed from the cell. So it's kind of a budding process. So it's not injuring the cell, it's just removing some of the membrane. If this is done over and over again, then it can be injurious to the health of the cell. But the cell can live a long time with viruses in it. Okay, does that kind of make sense? You can see lots of questions that could be asked for something like this. More or less information can be shown on the slide, and then you have to fill in the blanks. But here's a good example. These circled things are things that could be left out, could be left out, and you would have to say the pathway in between, or it could be shown, and then you'd say what happens between here and here, and here and here, etc. Okay. So questions about this one? It kind of gives you a whole encompassing thing on a DNA virus going into the cell. Okay. All right. So here is another picture. This is just kind of a shortcut picture. It's just telling you step 1, 2, 3, 4, 5. It gives you the different steps that we're doing, but what this is talking about is when you're an envelope virus, you can be persistent. You can get out of one cell, you fuse with another cell, you bud out of that cell. So as this persistence, you can be doing this to the cell for a long time. It weakens the cell, but it doesn't really kill the cell right away. It will eventually, but not right away. However, if you're a naked virus and you go through this exocytosis partial thing, you're usually lysing the cell. You're usually actually busting out of the cell and lysing it. So the envelope viruses are a little bit more persistent. They stay around a little bit longer, infecting cells, and the cell also lives a little bit longer. And the naked viruses, they are a little bit more brutal. They go through more of a lysing methodology. It won't kill the cell the first time, but over time it will. Okay. Questions about this slide? All right, so here's this first slide I showed you. This is for like bacteriophage. Come in, synthesize stuff, and then bust out. You know, and then that's it. That's the life of the cell. Here we go through it, we synthesize, we assemble, and very slow release. This you can see the whole life of the cell and stuff is going to be a lot longer, and the whole time frame is going to be a lot longer. This time frame is a lot longer only because even for animal cells, it takes almost 24 hours for the viral infection and replication and assembly. It could even take a little bit longer than that. Okay. So that's just comparing between bacteriophage, 25 minutes, and time. They don't give us time on here, but we know this is probably in hours. Okay, so this is in another section of the textbook. This is on HIV. So this is in chapter 18, and we will come back to HIV and talk about it. But HIV, again, it is going to attach just like a regular DNA virus would. It's got receptors, and remember the CD4 receptor, and it's got some co-receptors. So as it's making its target, it's going to actually be coming into the cell. Now this shows the whole virus itself coming in. For HIV, usually it's left on the surface right here. So you can see the part of the surface that it's going to fuse with, and then only this part in here is going to be released inside. So this is called the uncoding part. So once we're inside, then the genetic material is going to be reverse transcribed, etc. It comes in as a RNA virus, and then it brings in with it a reverse transcriptase, so we can reverse transcribe the RNA virus into a DNA. And then once it's reverse transcribed, it will then be led into the nucleus. Once it goes into the nucleus, it integrates into the genome. So it has an enzyme called integrase. It will help it integrate into the genome, and now it is for life in this particular cell. Okay, and if it does that to multiple cells and stuff, the person who has this has it in either their T cells or their macrophages, and it's for life until that cell is killed. And of course, you can't kill everyone's T cells because then they have no immune system left. And we'll talk about the immune system when we get there. Okay, and so again, when it's time, and they don't always know what triggers the HIV to decide to come out of the genome and become a virus again. But when it does, it has to come out. It's going to make all its co-proteins. It's going to do everything. It's going to assemble, and then it's going to package itself up, and then it's going to bud out of the cell. Yes?
That means HIV is considered only lysogenic?
It's not lysogenic may not be the right term. It's a pro-virus. That's probably a better way to look at it. It integrates into the genome. A lysogenic one means that it would actually come out and then bust out of the cell, and this doesn't. It just, it's kind of like herpes virus and some of the ones that lay dormant. This is just laying dormant in the genome. But at any time, it could come out again. I mean, people have had HIV for years, 20, 30, 40 years, now almost 50 probably, and it's still in the genome, so they live with it. And there's lots of drugs that can be taken to tamper it down, and we'll also talk about that when we talk about multi-drug and how drug targets that it can be made. Okay, so this is a slide specific for HIV. Yes, so I'm glad everybody has that. So you hear in the news now that a lot of funding is going away for Africa to dampen down the HIV. We should be really concerned about things like that because, in fact, HIV is very, very transmissible. So if it does become another health problem, it just could cost billions and billions of dollars. So I hope that never comes to play, but it is, HIV is a bad news. Okay, so something to think about. Oh, so this is one little problem. You don't have a calculator with you, but I just wanted to remind you. This is very similar to what we've done before and how you would solve it. So how would I even start something like this? What formula would I use? So the thing that should trigger plaque-forming units, we have colony-forming units, right? Exact same thing. So I'm going to put up here colony-forming units per mil. So that's going to equal the, I'll put plaque-forming units per mil. So we'll put plaque-forming units. We read on the plate, whatever we had on the plate. We had so many. And then we have our the way that we diluted it, and we had a 1 to 10, right? We put 10 into 90, so that's a 1 to 10. That's our first dilution, our only dilution that we had. So we'll put our dilution over here. And then we had what we plated. And it says 5. So how many plaque-forming units do you have in 1 mil? Well, what we actually had on that first part up there is 10 microliters. We put it into 10, and we put that whole thing on a plate. So let's say here was our vial of the virus, and we took out of there 10 microliters, and we put it into 90. And then we took the whole 90 and we put it on the plate. So we took this whole 90 and poured it on the plate and spread it around. So for the actual virus that got spread on the plate, we had 10 microliters. Okay? And this, then, it is per mil, so we're going to have to do, there's 1,000 microliters in 1 mil. So this part right here is just getting us to our mils, and this is our dilution. So, and our new plaque-forming units up here was 5300 plaque-forming units. Okay? So you now, I mean, this is exactly what we did with colony forming units. So this is just one more use of that. And we're going to use this same formula again when we do things with soil, that will be counting things from soil. So the plaque forming units per mil, the number of plaque forming units that you have, the dilution that we had, and then how much virus did we actually put on the plate. Well, we put 10 microliters because we took our whole 10 over 90 little mil and we dumped it all on the plate. So any idea how much these would be? I know you don't have a calculator, so. Okay. Okay, so we're getting different ones. So what I'll do it with my calculator. Can't do it with my calculator because I don't have it. So if we flip this to the top, so if we flip this up, this would be 53000, right? If I just flip this to the top, so I'll get rid of this. And if I put 10 over 1,000 like this, and I get rid of these, I have another 1 over 1,000, over 100, and I would add 2 more zeros for this. So I think this is 1, 2, 3, 4, 5, 1, 2, 3, 4, 5, 6. 5.3 times 10 to the 6th.
Stupid.
Yes, so this is D. Okay, does it make sense? Okay, so again, you could have a problem like this. I mean, this is not out of the reach of what we've already done. We've done colony forming units. We've done this. It would be a simple problem like this. And the idea is we would be counting colonies on our plaques on that plate, and then we would come to a conclusion what they are. So, any questions about this? Does it make sense? It just reinforces something we've already learned about colony forming units. And I want to reinforce, after this time, we'll be doing a count on your plates on the Tuesday that it'll be the Thursday afterward that we actually see our plates. We'll be doing some counts like this, and then we'll leave that again, and then we'll go into something else a little bit later in the course and use the same formula for something else. Okay. And then here's a stopping point, but I'm just going to talk about cancer a little bit, and then we'll take that stopping point as our end point. Viruses can cause cancer. I don't know if almost everybody knows somebody who's had cancer for some reason. Having cancer because of viruses is really not talked about a lot. There's not always very many people who get a cancer just related to viruses, but it is a problem. And those cancers are sometimes masked because the cancer is caused by a virus, but it brought it into a specific area of the body that is then called breast cancer or kidney cancer or something else. So I think everybody probably already knows some of the terms, but tumors and viruses, cancer, uncontrolled growth, that's all that really means. It's only when that uncontrolled growth starts spreading do we really consider it cancerous. Normally we just say, okay, it's a tumor, we'll take care of it, we can excise it or something, but there's certain points when that becomes impossible, either from location of it or because it starts breaking off and spreading throughout the body. And this is another beautiful thing about the human body. We have these nets that your body recognizes something is amiss. They go, gee, this area seems to be getting a little larger than it normally should be. We're going to put a net around it. And it actually formats a net, a fibrous net around a certain area to try to contain that area. But of course, if it's cancer, then the cancer is just going to break through that and do it. But your body still tries very hard at every point of anything that's abnormal, it tries to fix it. And it usually does a pretty good job. So usually there's going to kind of surround some kind of a tumor with some kind of material in order to keep it in a state that is benign. Because once it starts breaking off and metastasizing, then it's not benign anymore. Then it's actually a malignant tumor. And so again, this is just saying it's abnormal cells that are invading other areas just from having this uninterrupted growth. And the role that viruses play at all in this, there are some very specific viruses that we have. So Burkitt's lymphoma, Hodgkin's disease, Kaposi's sarcoma. In cervical cancer, I didn't put that on here, but many cancers can be exacerbated by viruses or actually caused by viruses. So also, I mentioned that viruses will integrate into the genome of an organism, and when it integrates into the genome, it's really not particular where it integrates. It just says, oh, here's a good place to integrate into the genome, opens up the genome, and it gets in there. It may be in a gene that is supposed to turn off something, turn off growth signals, or maybe it's in a gene that's supposed to be sending growth signals. But whatever may happen is the virus that gets into a genome could trigger those two signals to be abnormal. And so maybe you're getting signals to divide when you shouldn't, or signals to not stop when you should be stopping. So that's usually how they attribute viruses to cancer. So oncogenes are genes that are not yet cancerous. So they're just potential ones. Our proto-oncogenes are really the potential ones. And then these are the ones that are that could be potentially cancerous if something happens to them. And here is kind of showing, this is like a normal state. Here's the proto-oncogene. It's not cancerous yet. Here's a gene that's a repressor. Here's our mRNA. It's working fine. We're repressing something, so we're not allowing a cancerous event to happen. And here's maybe the first hit. Maybe something comes in here, and this promoter was inserted by a virus. Maybe it brought it from another cell. And this actually promotes this proto-oncogene and starts turning it on. And then when we're actually taking our repressor, we can actually repress this. So still, we have no cancer. But maybe we get another hit down here in our repressor gene, and that's maybe from another virus that's integrating there, and then that causes us not to be able to repress the oncogene that's in front of it, and therefore then that goes to cancer. So this is where really usually they attribute this to viruses. You know, they bring in and they integrate into places that change the normal working of the cell. And latent viruses, I think all people have seen some of these. This is what like herpes, not herpes, but shingles looks like. So this is shingles, and this shingle region is just all these little sores, and they just really itch. So there's viruses, or there's material or vaccines and stuff that they can give you for those shingles. That's just from chickenpox from long ago. And is there missing some slides or something? Is that, yeah.
I was just checking one of my papers.
Oh, okay, I just thought maybe I missed some slides in there. All right, so these are laid dormant, and then they come out at whatever stress levels. And these are like cold sores and stuff, all kinds of things that we get that are really from latent viruses that are in the body. And all these things that are called a provirus, these are the ones that are integrated in the genome and stay in the genome forever. Okay, usually they don't cause any problems, they just stay there. It's never happened to me, but I have a lot of friends that have had the shingles, and they tell me how terrible it is, and I go, how did you even know you had it? They talk about the itching and all the stuff, but I guess not everybody, it doesn't come. And I had chickenpox when I was young, so I guess I should be susceptible, but I'm not. The cold sore things, I think everybody's run around that for some things, and these are latent viruses that are in the genome. Okay, and let's do this. I think we have just a few more slides after this. So benign tumor, what do we call it? Yep, I see all the blue. That's absolutely correct. I mean, someday maybe it will metastasize. You never know. And again, once we get to our immune system, it'll be really cool to talk about all the things that your body does to prevent things like that from happening. And that's why we always want to get home early and go to bed early and get lots of rest, because you want to have your immune system be as strong as possible. Okay, so I definitely wanted to get to this slide to kind of almost close off. So how do we even study viruses? We know that they have to have a host, and they kill their host, so it's not easy to study them. So one of the ways they do is, I mentioned these plaques over here. These plaques, when they get pretty large, you can just take them out with a pipette tip or something, and then this is all pure virus in each of these. Okay, and if you infect it with the same virus, it's generally the same virus. Maybe little mutations in there, but it's the same virus. So then you can study it that way. And it's not really great, but it's the best way that we have for bacteria viruses. And then you can actually make cells. You can take out tissue cells, you can grow them, and then you can infect them, and the cells will curl up because they're being infected, and then you can use them to study what's happening when you put a virus in. And you can grow more viruses, but then you have to suck that out and keep a transfer from cell to cell to cell about every 20 hours, because they will kill the cells pretty quickly. And then the last one is fertilized eggs, and this is where you can inject the virus into the eggs. Once they're fertilized, they have lots of yolk in them and stuff, and they will be able to have viruses survive for a long time. And that's how most of our viruses are now made. Now, of course, since this slide was made, we have mRNA viruses, and that's a totally different game than going through all of this, and probably a game changer for the future too. But so bacteriophage, we use plaques, and regular viruses we put in fertilized eggs, or we use mRNA. Okay. All right, and this is where we can stop. This is talking about the viroids. We have these couple slides, and I think I'll just leave this to you on your own. And we talked about prions a little bit before, so there's only 3 or 4 slides here, and I will let you just go through these on your own. That sound good? No? Yeah. Okay. So yeah, it's time to go and get some rest so that your immune system will be happy and functioning right.
Yeah, after spring break.
Next Tuesday after spring break.
I thought it.
The ones that were up here. So yeah.